Hello, my dear students, and welcome back. Your board exams are just around the corner, so we're diving into one of the most important chapters, genome technology. This stuff is a real game changer, so let's get you ready to absolutely ace it.
Just think about this for a second. If a book had a typo, you'd just correct it, right? But what if you could do the exact same thing with the code of life, our DNA? You know, this isn't science fiction anymore. It's happening right now, and you can be sure it's a super important topic for your exams.
So before we can even talk about editing or reading this code, we first need to understand what it is. We're starting with the most fundamental thing there is, the genome. Just think of it as the complete instruction manual for any living thing.
Don't get scared by the big word, Betcho. It's actually a very simple idea. A genome is just the complete set of DNA instructions. Yep, it's the entire recipe book for making an organism, whether that's a tiny bacterium, a plant, or even you.
Now, what's interesting is that this recipe book isn't stored the same way in all living things. In simple cells like bacteria, we call them prokaryotes, the DNA just kind of floats around in a region called the nucleoid. But in complex cells like ours, the eukaryotes, it's all neatly organized inside a nucleus, with a little extra in the mitochondria and chloroplasts too.
So, we have this massive instruction manual. But what good is a book without a table of contents, right? We need a map. And that's exactly what genome mapping is, creating a detailed atlas of our DNA. Let's look at the two main ways scientists do this.
Okay, so there are two main types of maps. First, there's a genetic map. It's like a rough guide. It tells you the relative position of genes, kind of like saying Delhi is north of Mumbai. But a physical map? Oh, that's much more precise. It's like having the exact Google Maps pin for your friend's house in Delhi, giving you the actual physical location on the DNA strand.
So, how do we make that super precise physical map? Well, a key technique uses what are basically molecular scissors called restriction enzymes. Just look here. These scissors only cut DNA at very specific letter sequences. In genome 1, that cut site is present, so the enzyme snips the DNA into two smaller pieces. But now imagine a genome 2, where that site is missing. The scissors can't cut, leaving one big, long piece. By analyzing the sizes of these DNA fragments, scientists can create a really detailed map.
Okay, a map is great, really useful. But what if we want to read the entire instruction book letter by letter? Not just know where the chapters are, but read every single word. Well, that's where DNA sequencing comes in. It's the process of figuring out the exact order of the bases, the A, T, C, and G in a DNA molecule.
And you won't believe how fast the technology for this has improved. The first generation was so slow and cost crores of rupees just to sequence one human genome. Then, the second generation got much faster and cheaper. And now, the third generation is so advanced, it's literally like reading a book in real time on your phone.
This is the latest third-generation tech, and it is just amazing. Look at this. A single strand of DNA is passed through a tiny, tiny hole called a nanopore. As each base, A, C, G, or T, goes through, it causes a unique little change in the electrical current. By measuring these changes, the machine can read the DNA sequence in real time. How cool is that? It's mind-blowing.
And now for the most exciting part. Something you might have even seen in viral videos on Instagram or YouTube. We've learned how to map the genome and how to read it. But what about changing it? Let's talk about editing the blueprint itself.
The technology that lets us do this is called CRISPR-Cas9, and this analogy is just perfect. It works exactly like the find and replace function in a document. This incredible tool, which actually won the Nobel Prize, gives scientists the power to edit DNA with unbelievable precision. This is the tech that's now being used to cure genetic diseases like sickle cell anemia, something we once thought was impossible.
So, the crucial point is, how does it work? Well, it has two main parts. First, there's a guide RNA. You can think of it like a search query that finds the exact spot in the DNA we want to change. Then, there's the Cas9 protein, which acts like a pair of molecular scissors that makes a clean, precise cut in the DNA right at that spot.
Let's break it down into three simple steps. First, the guide RNA finds the target. Second, the Cas9 scissors cut the DNA. And third, the cell's own natural repair system kicks in to fix the break. And here's the really clever part. Scientists can supply a new, corrected piece of DNA, and the cell will use that new piece to repair the break. And just like that, the gene is edited.
So, with all this power to read and edit genomes, what can we actually do with it? Well, one really amazing application is engineering proteins to give them new, useful functions. We can basically create superproteins.
For example, imagine you have a big mix of different proteins, but you only want to pull out one specific type. With protein engineering, we can do that. We can add a tiny tag called a 6-his tag to our target protein. This tag is super sticky to the metal nickel. So we just use a nickel magnet to pull our tagged protein right out, leaving everything else behind. It's a super clever way to purify proteins.
And this is another brilliant use. We can take a protein from a jellyfish called green fluorescent protein, or GFP, which glows bright green under UV light. We can then attach this GFP tag to any protein we're studying. This lets us see exactly where our protein goes inside a living cell, just by looking for the green glow. It's like putting a little GPS tracker on it.
Okay, bachon, pay close attention now. We have covered a lot. Let's quickly summarize the most important points from this chapter that you absolutely need to remember for your exam. This is the most crucial part.
All right, screenshot this if you need to. These are your 5 golden points. 1. The genome is the complete DNA blueprint. 2. Mapping is our GPS, with genetic maps for relative location and physical maps for the exact pin. 3. Sequencing is reading every single letter, A, T, C, and G. 4. CRISPR-Cas9 is our amazing find and replace editing tool. And 5. Protein engineering lets us create superproteins with new jobs, like that purification tag and the GFP tracker. Master these 5 and you're in great shape for this chapter.
And here is a very, very important question for your boards. This type of question comes up so often. After this, take some time and try to write out the answer for 5 marks. Use the points and the diagram we just discussed about the guide RNA and the Cas9 scissors. Remember, practice makes perfect.
And that's it for our revision today. I am so proud of you all for working so hard and learning this complex but fascinating topic. You now have a good understanding of the technologies that are literally changing our world. All the very best for your preparation. Go ace that exam. I will see you in the next video.